Hello and welcome to your Week 3 lecture, which covers the structure and function of cells in the nervous system. In this lecture, which will be divided into 3 parts, we're going to talk initially about Learning Objectives 2.1 to 2.5 from your textbook, basically getting our sense or wrapping our heads around the central nervous system, the peripheral nervous system, how neurons and glial cells fit into those two parts of the nervous system. We're going to describe the functions of supporting cells, the glial cells within the central and peripheral nervous system, talk about the blood-brain barrier, and explain neural communication through the example of the withdrawal reflex. In subsequent lectures, in Part 2, we'll cover Learning Objectives 2.6 to 2.10. In Part 3, we'll talk about Learning Objectives 2.11 to 18. So on to Part 1, focusing on the nervous system, a kind of a review, the neuron, supporting cells, the glial cells, and the blood-brain barrier. So you will recall that the central nervous system includes the brain and the spinal cord, everything encased in bone. The peripheral nervous system is what is illustrated here in blue and represents all other nerves that run in the body from the neck down to the feet. Figure 2.3 is a cross-section of a nerve. And a nerve is basically a bundle of axons, several bundles of axons wrapped together. And you can see here from this illustration, and I'm going to get the red pen so I can show you, each of these little stones is one axon. And so we have a bundle of axons times, 10 or 11 here, with blood vessels interspersed, and they are wrapped together in this entity that we refer to as a nerve. So there are 3 types of neurons. There are sensory neurons, motor neurons, and interneurons. The flow of information shown in this illustration goes as follows. A person sees a glass of water, right? You see this glass of water. A signal from the sensory neuron passes to the interneuron. So this is step 1, passes to the interneuron, step 2, and then the motor neuron of the hand, step 3, which extends to grab the glass of water for the purposes of drinking. So again, 3 types of neurons: sensory, motor, and interneurons. And they are essential for performing tasks like perception, learning, remembering, decision-making, and a whole suite of complex behaviors. So what are the parts of the neuron? There are 3 basic parts. The soma, which is the cell body, is here. The dendrites, and this is not a very good looking number 2, and the axon. So these are the 3 basic parts. Of course, there is a lot more to be said about the subcomponents. For instance, your book mentions and emphasizes a 4th part, the terminal buttons, which are right here. So the cell body, the dendrites, axon, and terminal buttons are of great significance based on your reading. So the soma is basically what contains the nucleus. Its shape varies depending on the neuron. The dendrites are the arboritic or tree-like branches that extend from the soma, and they receive information from other neurons through synapses, which are the connections between neurons and by which one neuron communicates with the other. The axon is this long, thin cylindrical fiber. In this neuron, it's actually divided at the end into two sets of terminal buttons. And it's important to note that information from the presynaptic neuron to the postsynaptic neuron. So let's say there's a neuron over here. There's its cell body, and its axon, let's say, is projecting here. So this would be the presynaptic neuron sending information, and the postsynaptic neuron is this neuron that we already had illustrated. Information flows in this direction. And from the synapse into the dendrites, through the cell body, and then down the axon. The one exception is within neurons themselves, there's exoplasmic, transport. And as part of exoplasmic transport, which we'll see in the next slide, there is anterograde and retrograde transportation. So not necessarily communication like with the nerve impulse that we were talking about, but there is transport along these microtubules from the terminal buttons back towards the cell body in one direction, that's the retrograde direction, and towards the terminal buttons in the other direction, the anterior grade direction. And this is basically material contained in vesicles that travels along the microtubules to and from along the length of the axon. So exoplasmic transport isn't to be confused with what is generally described as a unidirectional flow of information from the axon of a presynaptic neuron. Up here, there's a presynaptic neuron, to the dendrites of the postsynaptic neuron into its soma, and then down along the length of its axon to receiving neurons that become the postsynaptic neurons at the other end of the equation. So the cell structure of a neuron, in addition to the axon and the terminal buttons and the dendrites, includes the cell body that has some important components worth mentioning. At the center is a nucleus, and you can see the cytoplasm, basically the space inside the soma. So here's the nucleus, and that's where the DNA is. Let's see if I can write DNA. So DNA is basically the genes. The deoxyribonucleic acid is in the nucleus here. In the cytoplasm, we also have mitochondria, which make ATP, powering the functions of the cell. We also have some other structures that are important for regulating the functions of the cell, but really the glial cells take care of neurons. Neurons have very basic cellular components. They don't make a lot of their own energy. The glial cells provide them with sugars and nutrients they need. And so the basic structures of the neuron cell body include the membrane, this phospholipid bilayer, meaning that it's made up of two layers of lipids. The cytoskeleton, which is basically the internal structure made up of microtubules that keeps the cell body from collapsing, gives it its shape, and the cytoplasm, which contains the mitochondria that make ATP, and again the nucleus, which contains the DNA that makes up the genes and any enzymes that are needed to fulfill cellular functions. So I include this illustration here just to show you that on Pearson there are a number of really useful video demonstrations of key concepts related to cell structure and function. Now we're going to turn our attention to glial cells, which are the supporting cells of the nervous system. So neurons make up only about half of the volume of the central nervous system. They have a very high rate of metabolism, second only to the gut in terms of the amount of power that it takes to operate the brain. They are in an approximately 1-to-1 ratio with glial cells, which are the most important supporting cells. The glial cells are the glue in one respect that holds the brain together, both functionally and both figuratively and literally. So the metaphor, based on the important functions of glial cells, is not completely inaccurate. So there are two types of glial cells. They fall into the peripheral nervous system and the central nervous system. And you can see that some of the functions are the same. So oligodendrocytes form myelin for neurons in the central nervous system, whereas Schwann cells form the myelin for neurons in the peripheral nervous system. In the peripheral nervous system, we also have satellite cells that support the cell bodies. Again, Schwann cells produce neurotrophic factors. In the central nervous system, you will know from your reading that astrocytes, which look like stars, engulf and digest debris through the process of phagocytosis, that oligodendrocytes provide support to axons, producing again the myelin sheaths, and those nodes of Renvier that you read about. Microglial cells are the immune cells of the brain, and they act like scavengers. Their small size permits them to squeeze in between spaces in the brain, traveling to sites of injury as needed. And the epididymal cells are a fourth type of central nervous system glial cell that creates barriers between compartments of the brain, again contributing to the overall structure of the brain. So astrocytes are star-like glial cells. They attach the soma of the neuron and the outside of the capillaries, and their thin tendrils go out in a sort of U shape, which you can see here. The oligodendrocyte, again, is responsible for myelination in the central nervous system. As you know, axons are tube-like structures. The points where the axon is not covered and is exposed are marked as the nodes of Renvier in this instance. The axon looks, as it's described by your textbook, like a series of beads strung on a single thread. And really, the nodes of Renvier represent the spaces between the beads, and the beads in this metaphor are the myelinated axon segments. So this cross-section shows that the oligodendrocyte has a soma, right? It has a cell body in the middle, and then its many tendrils go out and myelinate multiple axons from different neurons. So these are all different neurons simultaneously. So in your book, you'll recall mention of the microglial cells being important for the inflammatory processes of the brain, and being an area where problems emerge from traumatic brain injury, particularly because the debris that develops from traumatic brain injury doesn't get properly cleared. in the case of individuals who get repeated injuries or hits or blows to the head. This illustration is one we'll talk about in class that also relates to the subject of neural law, and it's of the brain of Aaron Hernandez, who is a famous football player in the National Football League who suffered chronic traumatic encephalopathy from TBIs endured over the course of his NFL career, ultimately committed some murders that landed him in prison for life, and took his life. And post-mortem examination of the brain showed some really gross anatomical markers of CTE and TBI, which we'll discuss in class, that also represent indicators of microglial dysfunction. So the myelin, you'll recall, is produced by oligodendrocytes within the central nervous system and Schwann cells outside the nervous system. And this illustration, outside the central nervous system. So in the peripheral nervous system, Schwann cells myelinate neurons. Here you can see that a Schwann cell will entirely surround the axon of a neuron, wrapping multiple times. And in the case of multiple sclerosis, there's an autoimmune attack of myelin in the central nervous system that impacts adversely the functions of the individual. And we'll talk about that in later sections of the course. So the final thing I want to mention to you is this idea of the blood-brain barrier. So Paul Ehrlich's experiment from 100 years ago showed that we have this thing called the blood-brain barrier, through which the brain is protected from things that we eat, that we breathe. It's selectively permeable, in large part because of the workings of astrocytes in the brain that regulate the composition of extracellular fluid and its permeability. The area postrema is the region where the barrier is weakest, and this has some functional significance in that it allows our brains to induce vomiting in the event that we take in things that are noxious, that are in our body and can cause physical harm or even death.